morning to all. It seems like scholars like us, while in the journey to reach our goal, there can be a long way to the end. My strong belief is
My great pleasure to invite Dr. Sugunan, who is faculty in the Department of Zoology. He's a scientist, academic, violinist, and a good motivator. Violin?
I do play, but I don't know what's
So he's a biochemist by training, and a marine biotechnologist, and a zoologist. Very interesting. He serves as a scientist in various research institutes, including Rajiv Gandhi Center for Biotechnology, Sri Chitra Institute for Medical Science, and various other places. Oh, Federal Biotech Park, what is that? Is that local?
There's one which was started, but when the funding has come, it's not properly started.
Okay.
They put up a gate. 30 crores has been sanctioned by the DBT. We have to see how it's taken up by the Kerala State Industrial Development Corporation.
Very nice.
We hope something comes out of it. I'm a consultant there.
Excellent. He's an author of books on biotechnology and particularly interested in viral hepatitis and bioinformatics, and he's associated with this department. Thank you very much.
Thank you. That's quite interesting. I never thought that all this has come up in writing here. All right. So we had several interactions and talks here, and most of these were concerned with the structure of proteins. So this is a work which one of my biotechnology students did just now. I guide both in bioinformatics as well as in biotechnology. This young lady started with working on a particular aspect in eukaryotes, then took up that interesting data and tried to work it on prokaryotes. She has now finished her work and then preparing her thesis. I think in another 3 months or so she will be submitting her thesis. The question that I put up here is structure of DNA more important than its sequence? You always talk about sequence, genetic engineering depends on sequences. You take a sequence, put it there, and then assume that it's going to work. Now there are lots of gathering evidence which says that that's not always true. Okay? This is something which whenever you search the internet and say DNA structure or chromosome structure, is what you get as a hit that most of these people who take from internet depends on, right? So here it shows the cell and the genome is inside, safely in. DNA, as you know, is a very inert chemical. It doesn't partake in any reactions. It just carries information. It doesn't take the information itself outside the nucleus. It sends the messenger RNA outside with its information. You will never ever see DNA in any reaction. So it's an inert molecule. And then this particular structure, you don't find it in all aspects of the cell. When you look at a cell, you won't even find any of this in a normal cell. All right? This occurs only when it's just about to divide. And this is what is. Now, the interesting thing about the human genome is something which is familiar. You have 25,000 genes, estimate. Some people claim it's 20,000, some others claim 30,000, so in between it is 25K. 9,000 genes are for tRNA and rRNA. They also need genes. They are not expressed, but they have to be transcribed. It's only a transcription, no translation. So the rest is 16,000 which code for proteins. And the number of proteins that has been estimated is based on an immunological aspect. It's more than 100,000 proteins. So 16,000 genes for 100,000 proteins. And are all these expressed? So when you assume this is not a proper, what you can call, when you say especially for non-biologists, assume that this is normal. This condition of the chromosome can be seen only when the cell is just about to divide. All the way. And this, the chromosome structure, the DNA structure, how it is around the nucleosome. It's around the nucleosome. DNA is bound to the protein. The protein is not bound to the DNA. And this is a wrong depiction of DNA structure. It's not the way it is. Okay, the helical structure is different. This is the one. But in almost all the textbooks you'll find this particular. No, it's wrong. This is the way it should be depicted. You have a minor group, major group. That's what gives it the structure. And then you have all these proteins binding there. And structure, all those biologists will know this particular structure. This is a normal biological DNA. The difference between these two are only the compression, the structure. Can you make it this way? Okay, it can be done. Just increase the concentration of your solution. Tonicity. Can the cell do it? The cell does it. There are lots of reserves of ions inside the cell, inside the nucleus. So when there is a signal, that's how the DNA changes its structure from this to that. This is artificial. And then you have C and D versions also. This is also dependent on ions for its tonicity, for its structure. Okay? And another surprising thing is the DNA that is bound to this nucleosome unbinds itself and then binds again 250 times every second. Okay? So it's quite fast. Can't even imagine. 1 second is quite a long time for a biologist. So within 1 second, 250 times it unbinds and then rebinds again. And the moment that it unbinds, it is amenable for some other molecule to attach onto it. It has to happen because that is when a particular sequence, a promoter or any other signaling molecule has a chance to bind onto the DNA. Otherwise, the protein protects it. So, now if that is the case, so it binds and releases 250 times per second. And this is a conventional eukaryote gene. So when you say that, you assume that it's a gene, you have all this enhancer, promoter, and then exons, and then everything comes out, that you have a gene which is ready to be translated, transcribed, translated. Prokaryote genes are different, they won't have these introns, so they can also be transcribed. Now, the conventional cellular dogma, you have DNA, mRNA is transcribed, translated. But when does this DNA allow itself to be transcribed? It's not always ready for transcription. So just like in replication, a replication happens only once in the lifetime of a cell, just once, not more. If it happens more than one, then it's a cancerous cell. There are controls. It's controlled by the DNA structure. These are all recent findings, but this epigenetic control is what is called. External controls has been noted just from 1934 onwards. So as in replication, expression of a gene product requires a gene product. Using a careful word there. A gene product is not always a protein. It can also be tRNA and rRNA. Every time an mRNA is produced, it has to produce sufficient tRNA for that particular protein. It has to produce rRNA for that particular mRNA. See, this rRNA is the one which makes the ribosome specific to that particular mRNA. So just by producing an mRNA, it's not going to be translated. You have to have an mRNA, and that particularly you have this untranslated region, the guide regions, which has to be taken into the ribosome. That requires a ribosomal RNA. That also has to be produced. So it's a series of happenings which has to happen before a protein is translated. There has to be a signal. It just doesn't go about producing a gene product. No, there has to be a signal, just like replication. External signal. The signal comes, and then initiation of transcription requires structural changes in the DNA. Otherwise, the promoter sequence or the enhancer sequence is not amenable for activation by transcription factors or any other factor that is involved. Right. So this is achieved by the signal or its messenger. A messenger is also enough. And what can be a messenger? Even calcium ions, magnesium ions. These are common messengers that happen inside the cell. Okay. So most of these signaling systems ultimately end up by the production of or the release of these ions. But how are they specific? How do they know that only this particular mRNA has to be produced? We still do not know. The specificity is still not known. How can? Because many of the signals finally end up with producing or releasing ions. The specificity is still not known. And if the signal is a hormone or a steroid, so the signaling process is a huge amount of time will have to be needed to explain all that. The signal comes to the receptor. All signals do not have receptors. The receptor will produce some other messenger. The messenger goes to the nuclear membrane. The nuclear, they take another messenger. So it's a long process. But still, there are other types, hormones or steroids. They can just dissolve across the membrane, because that's also a lipid membrane. Goes into the cell, the cytoplasm, across the nuclear membrane, it dissolves and then goes directly to the DNA. A hormone like estrogen, it has receptors, alpha and beta. It can also bind directly to DNA. It can dissolve across the cell membrane, across the nuclear membrane, go directly and bind to the DNA at specific locations which are called hormone responsive elements. So when it binds there, it changes structure of the DNA. Okay, so that's why you'll find whenever you look into literature, you'll find that estrogen and testosterone, all these steroid-like hormones, hormone-like those elements, or something that mimics these activities, are related to several of the physiological outcomes, including cancers. So this particular lady, she is interested in this particular compound. I saw one of the papers also in this abstract. Someone has presented something in bisphenol A. This is a monomer plastic, common plastic. Okay, so it's a hormone analog. It's an estrogen mimetic. It mimics the activities of estrogen. So she was interested on a particular fish that survives on highly polluted water. It's called anabas. Very high concentration of plastic. This survives. How does it survive? So she started a study with that. You give it high dose. So when you bring it to the lab, then you do all sort of horrific things, which the ethical people do. But for fish, luckily nobody has put any ethics. You can do anything with a fish. And fish is a good model because anything you put into that environment, so water, gets absorbed, because they have what is called as a single circulation. Goes into the mouth, gets directly into the bloodstream. Takes into the gills, okay, goes directly into the bloodstream. And whatever happens, it goes directly to the liver and across all of these systems. It's a very good model to study such things. So what she found was in higher concentration, it turns the male fish into a female. Okay, and one of the senior students, they actually showed that in the testis there are ovaries. Alright, so then some albinos were also seen in the second generation of they maintained this fish for quite a long time. They're very sturdy fish, and the effect is concentration dependent. Toxicity, everything they do, all the LD50, this concentration, low concentration doesn't matter, nothing. Low concentration in the sense, you say 2.5 milligram per liter. That's a high concentration for us because it doesn't usually occur in the environment or in the water that you get bottled. So all this is concentration. Toxic and viral for the fish at 15 milligram per liter. For the fish, it's toxic and viral at 15 milligram per liter. And it's reversible. How is that possible? Okay, reversible. You remove all this, and after some time you take sections, including the liver, you find it's almost back to its old histological normal conditions. So she was interested. He's a biotechnologist. So fish is something new to her, because since we work in the zoology department, we put her across to that. So the question is, where does it act? Estrogen has receptors in almost all the cells. Only the ratio of the receptors between the cells, including males, have those receptors. So at the genetic level, at the proteinaceous level, physiological. Then the question also she asked is, how about prokaryotes? Well, if it's binding to DNA, will it also affect prokaryotes? And this, for a biotechnologist, more easy to handle. So she went on to that. And what she did was, she took this BPA, included in the medium, it's just simple your agar preparations, and then using that disk diffusion method, Kirby disk diffusion method, she prepared various concentrations, and surprisingly what was seen was at concentrations maybe reasonably higher, even at 15 milligram per ml, where it kills the fish, you will find that there was an inhibition zone. Later on, second day, third day, cells start growing inside. So he thought that it's a toxical tolerance. So several experiments were done. I just showing only one of them. She picked up these colonies. Okay, she picked up those colonies which are seen as tolerant. she designed a modified Amis test. You know what Amis test is? We test for carcinogenicity. You put this toxic material in the medium, you take in this colonies, allow them to grow. And she got colonies even up to 25 milligrams per liter of that particular medium. So these are resistant colonies. Okay, so then she designed a very interesting experiment. She took this, put them in LB broth, okay, and LB broth that contains BPA. The surprising thing was these colonies were not able to grow in a medium without BPA. Alright, so now you have taught them to grow in BPA, they are not able to grow without BPA. And this is the normal bimodal curve of E. coli. It's bimodal because LB broth has a little bit of glucose when it grows with the glucose first, and when the glucose is exhausted, some of the colony, the number comes down, and then it starts utilizing protein. Alright, so here this is the one which she calls as induced ones. That requires BPA. There is no bimodality here. This is a bimodal normal cultures. So then she hypothesizes that this particular colony is now dependent on bisphenol A for its food. And surprisingly, it's not published, but someone else also gave the same idea. So they interacted with these guys who are in Canada. They also are trying to work out the biochemistry behind that. Alright, so now the next question was, so if it's an estrogen mimetic, it should work in eukaryotes. How about prokaryotes? It should be binding to DNA. So one of my colleagues was talking with me in the college one evening. She was also overhearing, it seems they bought a new FTR machine. Okay, good. Then I thought, why don't you do some DNA analysis just to add some good quality molecular work. So she gathered all his DNA, fresh DNA, ran an FTA, this is a double stranded pure DNA, and this is bisphenol A. Put them together. Okay, so this is in vitro. Put them together, and so both together, this is what you should expect, but then this is what she got. So her interpretation, I'm not an expert on FTIR. We took it to some other guy who said that in our own chemistry department, so they say there is an interaction but no reaction. This is in vitro. Okay, there is an interaction but no reaction. That means it somehow attaches, maybe intercalation is occurring. Alright, that is in vitro. Now, she did another experiment. She took the DNA from those bacteria which have been treated with BPA and which are growing in BPA containing medium. And this is what she got. Okay, 5 milligram. And funnily enough, these FTR guys say that these are peaks. I don't know, in some they say as bands, okay, whatever it is. So 5 milligram, not a big problem, no. When it crosses that, there are changes in the structure. And in 50 milligram, the bacteria do not survive enough. But remember, if I try to grow this bacteria in directly to 50, a fresh colony of bacteria which has not seen BPA, and put it in 15 milligram, in a medium containing 15 milligram per liter of BPA, they die off. An induced cell which is able to survive in 2.5 or 5 milligram, you give it then there is no problem, it just grows there. What is the difference? So she proposes that there is some sort of an interaction with the DNA structure. So I am throwing a problem to bioinformatics people. Can you work out or give a little more stress or interest on DNA interactions with these factors which are also called as epigenetic factors. You will be looking for proteins, you will be looking at gene sequences, and you say that this is going to be expressed. No, it's not going to be. If it is hidden, okay, it has to be open. If there is a factor which keeps on hiding. You have lots of pseudogenes. I will be carrying several pseudogenes for hemoglobin in my body. Everyone has it. It doesn't get expressed. Why? It's not exposed. Or it doesn't have a promoter-second substrate. Even if it has a promoter-second, if the promoter-second is not exposed, there's no point in having the gene. Okay, you look for bioinformatics, you say that this gene is there, you say it's going to be exposed. No way. Okay, so yesterday I had that interesting talk about your hardware. So it's already, yes, you are born with those genome hardware, but the cell is going to modify it. It has ways by which it can modify. That's what she showed. And then she says there's a biotechnology. So now the picture, she had a biotechnology, that's what she wants. Can she use it for bioremediation? Maybe she can, but the thing is, bisphenol A will not be freely present in the system. Okay, it's a monomer. So there some other interaction has to be there. But then if you have this bacteria, it can digest bisphenol A. And the fashionable word that we have, metabolic engineer or metabolite engineering. You use the toxic to adjust the metabolism so that it's using the toxin as its own food. This is not something strange. There are several such things in even in humans, in mammals, even for cyanogen glycosides. Okay, cyanide. It's a long story. So epigenetic. So these are what are called as epigenetic modification. They involve small chemical molecules that bind to DNA and expert their effect by modifying gene expression. It's modifying gene expression. It's not modifying the gene. There's no mutation. Nothing. It's just modifying expression. And there are several different types of modification, but the best is the methylation. This could be methylation. FTR says that it could be methylation, but both, it also seems to look like hydroxyl. But if you see the bisphenol A composition, then both it has OH as well as cis. And it's a very interesting molecule because you flip it from it, whatever it is, it has the same structure. Right? So, attach them to specific on DNA, specific sites on DNA. Do they do it? That's the next question. Okay? And then it causes, this is not BPA interacting with DNA, but I just took it from there just to show you what should happen. So it is just intercalation, just like Ethereum bromide. It's there within the group, and then it's changing. The structure of the DNA is being changed. Okay. So now what's the application? This was from a paper in 2003. It says food or the lack of it influences the inheritance of epigenetic modifications, with 2 examples being aracanets at 2003, put in by Nature Genetics Center. Aracanet and famine or obesity. Okay. So they function as epigenetic factors which are transmitted to offspring. Alright. So when does it affect? When that particular person was a child in his or her mother's womb. Now there are proof to show that. Okay. So children born to these people are likely to develop disease as well, even if they themselves have or will never chew areca. Alright. Now the proof, just few months back, I forgot the exact volume, Nature had published one. Your grandma is responsible for your diabetes. Your grandmother, not your grandfather. Okay. It seems as a fetus, your germ cells were produced when your mother was a fetus or a father. Okay. So that was some factor would have made that, because at the embryonic stage up to 14th day it's not differentiated. Epigenetic influences can be there. It goes along with the DNA. So the cells, when they differentiate, already their germ cells will also have those epigenetic factors. Alright, so very convincing evidence has been given in that particular. Unfortunately, when you go to the library, you put it in a paper, get a paper, write it down, and then the volume, everything is forgotten, you put it somewhere. So I know that there's a paper, it's either September or October, Nature has this. Okay, which says this proof for that. Your grandma is responsible for it. Okay, so the nature-nurture controversy, okay, that shaping your minds and bodies from the point of view of nature and nurture, both together, but working together to provide the best chance for immediate survival. So that was when I heard that talk yesterday. So hardware being modified by a cell. You cannot change your genome, you can modify it. So it's grandma's role. Mechanism by which these carrier ingredients is extraordinarily unique. They are transmitted to offspring in the form of chemical modifications at the time of a fetus. So a fetus, its own cells plus next generation cells are also being formed at that particular time, which is also affected by the epigenetic factors. Okay, so it's a rigidly configured hardware which is modified by epigenetic factors. So that's the question I want to ask you. Can you show more interest on that? They do occur normally, several cellular processes, differentiation, providing mechanism by which you are able to adopt it. Okay. So now the question is, what Lamarck said, can it be true? Because if you go back into science history, Darwin never denied Lamarck's theory. He said it's possible. Now it shows that possible. Okay, at that time, people they did experiments by which they cut the tails, but this is not that case. You are not deleting something. This is something which is added by the environment. So when that comes into the population, that's what is seen in the microbial experiments. Okay, there are several cells colonies, but only few can grow within that particular location where the concentration of BPA is toxically high. It's not a mutation. Some genes have been activated, already present. Okay, so when you have that in the population, then we can see that this particular population is the one which is alive, which cannot survive without BPA. Okay, so if you look at it the other way, all this pollution, all that, maybe a few generations, the human a few generations down the lane, they will be able to live only if the air is polluted, right? Okay, so the applications, induction of uncharted paths, nanomedicine. One of my PhDs after doing a she is now the head of the department of nanotechnology in neuralism. It is surprising. She asked me one morning, see, there's an offer here, can I do it? Okay, so just jump into it. We don't come with everything in our brain when we are born, right? And within 3 years, she is now the head of the department. She says nanomolecules can be designed so that in such a way that planar, or it just slips through the membrane and then goes into the nuclear and then binds to DNA. Very interesting aspect. So, nanomedicine, medicine that directly acts on the DNA, not any protein which is interacting with several other hubs. Okay, it just goes to the directly and binds to the protein. Okay, and then Ayurveda does that. Many of these preparations, Ayurvedic preparations, they will either have something ionic, including mercury. It's a delivery system, or oil. See, when you have something in oil, it's a missile which goes. I'll just finish. Okay. See, one problem with the teaching process, when I hear the bell, I stop. Otherwise, we don't. Yeah, you start with the bell and then you end with the bell. Okay. So, the lipid coating enables it to cross across the membrane. So, Ayurvedic prepares most of these have oily nature, which is allowing the cells, this material to get into it. All right. So, thank you.
Fascinating. Do you think it's your grandmother responsible for that response? Okay, questions please. Pavan. You have to ask. Any questions? Comments?
Very interesting, very interesting.
Thank you.
Sir, you have shown that bisphenol can bind to DNA. Like
Yeah.
Is it a computational study?
No. It's actually FTIR.
Okay. The proposed model is based on the computational study.
Yeah.
That's
one symbol. It's just saying that it could be possible. It's not.
Okay. So my doubt is. This contain two benzene ring, two rings with a hydroxyl group, benzene ring with a hydroxyl group.
Yeah.
So if it is a molecule, if it is a planar molecule only, then it can be entered into DNA. I mean, intercalating with the DNA. So several other molecules like taccharin, one is example. It's used in Alzheimer's disease. The off-target interaction is there by binding to the DNA. So have you checked the energy of this?
No.
Because there is the chance is I don't know whether I am right or wrong, but the chance is very less, I think, to intercalate the DNA.
Okay. All those questions I have thrown it to you because this particular lady, her husband got transferred to Nashik. She is now in Nashik. She is not going to continue this work. Anyone here who is interested, are welcome to take it up. I mean, it's quite interesting. It's at particular stage. See, in my case now, I work in a place where it's not very. We don't have so many instrumentation. So the students who come to me, I give them the choice to work on whatever they are interested in, whatever is doable. All right. If I have to take it further, then it has to be FTR studies, crystallography, and then that particular DNA interaction study, which has to be clearly and well done with evidence. Okay? So, if anybody is interested, that's why I said somebody has worked on bisphenol A here, you can take these ideas. Even not just this, I told you about cyanogen glycosides. This is something which you find in tapioca, the bitter taste in tapioca. My parents used to say you should not eat tapioca, especially those bitter ones, in an empty stomach. You try to eat it, you feel giddy because it has cyanogen glycosides, goes into the stomach, reacts with the hydrochloric acid, releases cyanide, makes you giddy. It kills, right? Have you, I don't know, at least Keralites will know that the kid, the goat, the young one of a goat, which eats tapioca leaves, dies. But feed it with tapioca leaves, little, and then after a few months, it can eat tapioca. Alright, that is whenever we have this tapioca, especially this one we have is M4, with the CTCRI people develop one with high concentration of cyanogen glycoside. The intention was so that it's bitter, so the bandicoots, the rats, they won't eat it. Unfortunately, this particular cyanogen glycoside is also found within the starch. So they say if you are cultivating M4, if you are going to eat M4, you boil it, drain out the water twice to remove that. Otherwise, this cyanogen glycoside goes into your liver and then it can create havoc. But that was a study I did when I was in the medical college as a biochemist there. There are certain people who grow this here in a particular locations and they have neural problems. They eat a lot of this particular tapioca. In their liver, there was an enzyme, higher concentration of an enzyme which is called as linamases, okay, which is not normally seen in other people. So this particular chemical which is in the tapioca goes to the liver and induces some gene which is already present there. If it is not present there, how are you going to have this particular enzyme expressed? Right? So epigenetic induction. So it's vast, it's beautiful. Not just proteins. Go for DNA structure. Yeah.
Thank you very much. Thank you. Let's give him another round.